Warm welcome from my side here, calling in from Cologne. I will give you a brief rundown of basically the tools that we as MSAID develop and that are now also integrated in Proteome Discoverer. I will discuss what they are and why you might be interested in them. And I hope to be able to answer your questions at the end of the seminar, if you might have any. Just for clarification, I wasn't actually involved in the project that generated the libraries. That were other people that are now also part of MS8. I just want to state that for clarity. I was more focused on the biology side, but I'm now firmly into the bioinformatics and am now basically sort of managing our efforts here at MS8. MS8 stands for Aiding Mass Spectrometry with AI, and our goal is to basically replace the heuristics that most of us have been using for the past 25 years with artificial intelligence in order to make them more sensitive and more comprehensive. And our tools that we've developed so far, there are 3 of them, and they're called Inferys, rescoring, and Chimeras. And all these 3 tools are integrated into Proteome Discoverer. Inferys and Inferys rescoring since version 2.5, and Chimeras, as Michaela mentioned, since version 3.0. I will, throughout this presentation, go over these 3 tools. I will describe them, describe their benefits, and how they interact in Proteome Discoverer to give you access to powerful AI for your data analysis. And we'll start with Inferys right away. So this is a slide that I very frequently show when I sort of discuss the merits of why would we even use artificial intelligence in order to analyze our data. So here, this is a sketch of a typical MS2 spectrum, and the way traditional search engines would analyze such a spectrum is they would take all peptide precursors that would potentially fit this particular spectrum. They would calculate all theoretical fragment masses, and then they would simply count the presence and absence of a theoretical fragment, and then calculate a score based on those. However, as we know, peptides fragment in a specific way. So certain peptide bonds are more likely to break than others, and hence, when we look at mass spectra acquired of the same peptide on the same instrument, then we typically see a conserved pattern in fragment ion intensities. And using these fragment ion intensities really is key in order to increase sensitivity of your data analysis workflows. And traditionally, that has been done using spectral libraries. These tools are widely used, and there's one also integrated into Proteome Discoverer. But spectral libraries, of course, have limitations. They do let you interpret intensities of fragment ions, and they allow you to calculate spectral similarities based on these intensities, which increase your sensitivity. But you have to acquire them. You have to acquire a spectral library, preferentially one that really fits to your sample that you're currently analyzing, that has been shown in the literature time and time again. You can, of course, also work with bigger libraries, but you will have some drawbacks associated with those. And I won't go into too much detail there. But what we really want to do now, and what we can do now with the advent of AI, is instead of acquiring a spectral library, we can actually predict them based on the peptide sequences that are contained in a FASTA file. So machine learning really is the driver behind all of this, and machine learning needs one thing and one thing only in order to work properly, and that is a huge amount of training data. During my time at the Küster Lab, there was one particular project called the Proteome Tools Project that generated huge amounts of high quality data on synthetic peptides. And that is a basis for training neural networks. And we use this data also to train Inferys, which is our neural network that can predict physicochemical properties of peptides, among them the fragmentation behavior. So how do we do this? We gather high quality training data, such as the Proteome Tools data, but also other data from Pride. We select spectra that are very well representative and are of good quality. And then we have a so-called AutoML pipeline where we train thousands of different models on these spectra and select the best performing ones automatically, convert them into an appropriate representation to be used then in Proteome Discoverer or other services that we offer as MSAID. And when we do that and we use one of these models in order to predict the fragmentation behavior of a peptide that we see here on the right-hand side, this is a typical result that we usually obtain. So up top, we have the experimental spectrum, and on the bottom, you have the prediction of Inferys for that particular peptide sequence. And as you can see, the intensity distribution of the B and Y fragments is virtually indistinguishable, and that is reflected by two key quality metrics that we use to measure spectral similarity, and that is the spectral angle and the Pearson correlation. This here is a Pearson correlation of 0.99. So the predicted spectrum here at the bottom is virtually indistinguishable from the experimental spectrum up top, which basically means we can use these fragment ion intensity predictions as priors in order to answer the question, is this experimental spectrum that I now acquired similar to the prediction that I made? Yeah, so Inferys does that. Inferys is a neural network that is capable of predicting fragment ion intensities among other physicochemical properties. We also predict retention time, but I won't go into too much detail here. Feel free to ask questions related to retention time predictions after the webinar. But in essence, Inferys has been part of PD since version 2.5. It can be used to predict spectral libraries that can then be used with, for example, MS-PEP search in PD in order to analyze data. We upgraded our model in 2022 to Inferys 2.0 with and included basically charge 1 precursors in our training data set, improving prediction performance there. And our current inception that will be part of PD 3.1 is Inferys 3. That now supports any protease. It supports peptides between 7 and 30 amino acids in length, charges 1 to 6. We support CID and HCD as fragmentation methods, and now also support phosphorylated peptides, which I think many of you will like. As well as modifications that we were able to support previously, such as oxidation and carbamidomethylation. In terms of labels, we do support TMT and TMT Pro, as well as SciLec. And I just want to highlight one of the new key features of Inferys, and that is the capability to perform prediction of phosphopeptides. And we also included there fragment ions now from neutral losses, and they are very helpful in explaining even more experimental intensity than we were able to do with previous inceptions of Inferys. So here's a typical experimental spectrum of a phosphopeptide. The sequence is annotated here up top. We labeled Y and B ions in blue and orange respectively. And then we have the prediction here in green at the bottom. And you do see that we now predict water losses, we predict phosphor losses, ammonia losses. And all of these losses basically amount to the fact that we now with Inferys 3.0 on average can explain 50% more intensity than we were previously able to explain only based on B and Y ions with Inferys 2.0. So this is really a major upgrade of our machine learning model. And I'm sure you'll appreciate the accuracy of the predictions, both for spectral library prediction, but also other approaches that you can do with Inferys and the predictions that it provides. So now you might be asking yourselves, well, which prediction? What can I really do with these predictions? I mentioned that you can use spectral libraries with MS-PEP search, but one other tool that we at MSAID develop and integrated into Proteome Discoverer is called Inferys rescoring, and basically makes use of the predictions from Inferys in order to rescore the results of sequenced HT. In the name is already encoded how the workflow looks like. This is a typical workflow as you would use it with Inferys rescoring. Here on the left-hand side, we have the different nodes. There's nothing new really here apart from the fact that you slide in Inferys rescoring in between sequenced HT and percolator. And the general approach here is that you start with an experimental MS2 spectrum. The spectrum gets scored by sequenced HT, and not only the top rank, but multiple rank hits that sequenced HT evaluates are then passed to Inferys rescoring. This uses in the back end our Inferys model in order to predict fragment ion intensities for that particular candidate peptide. And then we calculate a whole host of intensity-based figures of merit that then get added to the results of sequenced HT. And based on those, we can better distinguish true positives from false positives. One key feature of these intensity-based figures of merit, such as, for example, the spectral angle, is depicted here on the right-hand side, and that is that they are much more stable over the chromatographic peak. So here we have an XIC of a typical peptide, and we plotted the intensity on the y-axis and shaded the area. But what is key to see here is that we also plot the relative cross-correlation score, the relative number of matched fragments, as well as the relative binomial score, in addition to the spectral angle across retention time. And I'm sure you can appreciate that the spectral angle really is more stable across retention time, giving you that increased sensitivity for identification, even when you have very little peptide eluting from your column at the given time when you generated that experimental MS2 spectrum. So what do we recommend Inferys rescoring for? One thing we recommend it for is to boost the number of identifications in immunopeptidomics analyses. So here we have a slide where we compare the performance of sequenced HT Inferys rescoring with Inferys 1.0 and Inferys rescoring with Inferys 2.0 in terms of its capabilities or their capabilities of identifying PSMs and peptides in a particular data set that we depicted here. We took that data from the study here. And as you can appreciate, as soon as you use rescoring and add these intensity-based figures of merit, you can substantially boost the number of identifications, both at PSM and peptide level. That's a 68% boost at the peptide level, which really allows you to dig deeper into these HLA data sets and pull out potentially new neoantigens. But you might ask yourself, well, are all of these additional identifications really true? Well, for this particular study, we took a look at sequence motifs that are apparent when one looks at the identified peptides from a given sample, and these are basically cell lines that were engineered to express only particular allele. The allele is depicted here on the y-axis, and that has a specific motif associated with them. And now we separate the peptides out in three groups. So peptides that are identified only by using sequest, that are identified by using rescoring, or that are unique to rescoring. And as you can appreciate, the IDs that we add with rescoring, they basically reflect the sequence motif that we expect for this particular HLA allele. And that strongly suggests that these are actual true HLA peptide identifications. So HLA immunopeptidomics in general, Inferys rescoring works beautifully there. Where it also works very nicely is when you do, for example, ion trap-based single cell data analysis. So here we took another data set from a publication. The citation is here at the bottom, and we looked at the performance of sequenced HT alone versus sequenced HT with Inferys rescoring. And as you can see, if we look at the number of identified PSMs, peptides, and protein groups in all these categories, we boost the number of identifications in the double digits, which is a huge gain when it comes to identifications if you're working with single cell data. And where do these gains typically come from? Well, they come from the fact that, in particular, ion trap data is or can be very noisy. You have to use very wide fragment ion tolerances in order to analyze that data. And this particular identification here that was basically above Q-value threshold when you only consider the the features that sequenced HT calculates. This identification can now be promoted to be a true identification below your Q-value threshold, so you can actually confidently identify this now when you use rescoring, while previously you weren't able to confidently identify this. So again, a big advantage there, and really not much difference in terms of workflow. The only thing that you have to do is slide Inferys rescoring in between sequenced HT and percolator and benefit from its increase in identifications. And with that, I'd like to switch gears yet again and talk about the elephant in the room, so to speak, Chimeras, and its capabilities when it comes to the data analysis of all types of data, and the AI-driven analysis of all types of data. So, what is Chimeras? So when we started MSAID, we realized that there's one particular problem that that really plagues all of mass spectrometry-based proteomics, and that is the presence of chimeric spectra. And typically, when we think about this problem, what we use to visualize it is this picture here, where we plot the contents of a spectral library containing roughly 400,000 precursors, and we plot just the number of precursors in bins of 2 Thomson by 1 minute across the entire precursor MZ and retention time range. And what you can see, each and every single one of these 2 Thomson by 1 minute bins, they will contain multiple precursors, meaning that even in a typical DDA measurement where you have very narrow isolation windows, you will have multiple precursors isolated and fragmented together, generating chimeric spectra, which consists of fragment ions from multiple peptides. This is covering the DDA part, but with wide window DDA or wide window acquisition or even DIA, we generate these types of spectra on purpose. So we have to devise a way to tease these different peptides that are contained in a single spectrum apart from one another. We are not the first ones to realize this problem, of course. That problem has been tackled in the past with various different approaches. 2 of them I showcase here. We call them the subtraction and the duplication approach. One approach, the subtraction approach, would identify a peptide in an experimental spectrum, would then remove the peaks that belong to that identification from the experimental spectrum and submit the remainder of the peaks to the search engine, again with a different precursor mass. The duplication approach, on the other hand, what it does, it duplicates the MS2 spectrum for each precursor in the isolation window and submits these copies of spectra separately from one another. But both approaches have a fundamental drawback, and that is that they either use information too often in the case of the duplication approach, or not often enough in the case of the subtraction approach. When we realized this, we went to the drawing board and devised a new AI-driven way to deconvolute chimeric spectra. And essentially what we're doing is there's nothing magic about the way we deconvolute the spectra. There's no AI in the deconvolution per se. That's just linear algebra. We just use the predictions in order to make use of the intensities that we observe in the experimental spectrum in order to tease them apart. And when we do this, we basically can take an experimental spectrum like this and identify not only one peptide in this experimental spectrum, but 2, 3, and even 4 different peptides in that particular experimental spectrum. And we do that by basically minimizing the number of peptides that best explain the experimentally observed intensity. Yeah, and when we do that, you can see that the experimental intensities and the predicted intensities here, also for the shared ions, basically very much align, suggesting that these are true identifications. And if you look at the predicted retention times of these 4 peptides, you see that they are all supposed to co-elute given our prediction. And if we look at the corresponding isolation window in MS1 that generated this particular MS2 spectrum, you see that all 4 precursors are present and were in fact isolated, co-isolated together and generated this chimeric spectrum, which we were able to successfully deconvolute using this method. In the process, we also calculate what we call contribution of a peptide to the experimentally observed total ion current. And that will be important later when we talk about quantification as well. So keep that in mind. I will reference this at a later stage. So how does Chimeras take advantage of this new capability? Basically, Chimeras consists of 4 steps. We have the first step is a first search where we do intensity-based scoring of using predictions. We then recalibrate our machine learning models. We perform deconvolution of MS2 spectra in a second search, and then afterwards we perform error estimation using Mockapot. And Chimeras, as it's integrated in PD 3.1, now will support basically any acquisition method. So it will out of the box support the analysis of DDA, DRA, and PRM data. You'll be able to analyze data from samples that were digested with any protease. You'll be able to analyze label-free TMT, TMT-Pro, and Silec data, and also phosphorylated peptides. So what is the benefit of using Chimeras? Well, Chimeras works very well in deconvoluting chimeric spectra and hence can really boost the number of peptide identifications. And in typical DDA datasets, what we see is that we double the number of peptide identifications in comparison to sequence HT. Here, this particular dataset is a 1-hour HeLa lysate where we compared sequence HT versus other tools along the sort of different generations of Proteome Discoverer, one being sequenced with precursor detector, the previous way of Proteome Discoverer to deconvolute chimeric spectra, basically, that uses the duplication approach. Then Inferys rescoring and Chimeras. And as you can see, at the number of PSMs, we add 388% to that. We double the number of peptides, and we add 50% more proteins to the identification list when we use Chimeras in comparison to other approaches. But if you're not after really sequencing your sample in great depth, but you're after throughput, Chimeras can help you there as well. Here on the right-hand side, we have a plot where we analyze the same sample using different gradient lengths. And this plot is simply there to depict that if you don't care about the number of identifications, but you rather would like to shrink the time that it takes you to arrive at a set number of identifications, Chimeras, or analyzing your data with Chimeras, will allow you to basically cut your measurement time by a factor of 3. So previously with sequence HT, you were able to identify this number of peptides in 46 minutes of gradient time, so roughly 18,000, 19,000 peptides. And now we can do the same number of identifications in a third of the measurement time. So really boosting the throughput there. If you are curious how Chimeras compares to other search engines, we have results for that too. We took data from a public data set again and analyzed it with various different search engines, among them MS-Fragger, MS-Amanda, MaxQuant, Comet, so the usual suspects. And Chimeras basically outperforms all of them by more than a factor of 2, apart from MS-Fragger, but the gap is nearly a factor of 2 in comparison to MS-Fragger as well. So you can really dig deep into these DDA data sets with Chimeras now. And the reason for that is that these intensity-based figures of merit really allow you to make an identification even if you have only very few peaks to come by, because you can judge whether the intensity of these peaks are what you expect them to be. This is visualized here on the right. So you see that the number of shared peaks for peptides that only Chimeras identifies in the data set here on the left is shifted towards lower number of peaks, but the spectral angle is pretty high for these peptides still, with a spectral angle of 0.75 that equates to a Pearson correlation of about 0.85, I believe. So we are really certain that these are true identifications that we can only make due to the fact that we have now intensity as another dimension that we can look at in our experimental spectra. So this covers the DDA side. But we now added with Inferys 3.0, if you remember, also the capability to predict phosphorylated peptides. So how does Chimeras perform on phosphorylated peptides? Well, I brought data for that too. Here's the Chimeras workflow on the left again, just to visualize it, that the workflow is now much simpler. You only have the spectrum files, the spectrum selector node, and then Chimeras. And here we have data on the right-hand side from a study delineated here on the at the bottom, where we can really see that Chimeras, in comparison to Sequest and Comet, really boosts the number of identifications of phosphoserine, phosphothreonine, and phosphotyrosine peptides. And the good thing is that with Chimeras, we actually cover most of the identifications that these other search engines were able to obtain. So the overlap is substantial. And one big other advantage is that in this particular workflow, when you're working with DDA phosphoproteomics data, you can even use the PTM-RS node to localize your phosphorylation sites. So here we plot now the unique site count, and as you can see on the unique sites, Chimeras also outperforms both Sequest HT and Comet. With that, I'd like to talk about what I think is one of the coolest features of Chimeras, and that is that you now have one search engine, one algorithm that allows you to analyze any type of acquisition method with the very same algorithm. So DDA, DIA, and PRM data. And for that, I would like to briefly circle back to what I mentioned earlier about the contributions that we calculate. For each precursor, we basically calculate what its contribution is to the experimental MS2 spectrum. And we use these contributions as a means to quantify how much precursor we have in a given MS2 spectrum. So we take our deconvolution approach, we arrive from a chimeric spectrum to the contributions of the orange and the blue peptide, and the contributions we then trace along retention time in order to arrive at MS2 level quantification. And this is done using the fragment ions quantifier node. And basically, what you can think of, the way you should think about these contributions is that it's the explained, the amount of explained intensity that can be attributed to that particular peptide in a given scan. And if we trace these contributions then along the gradient, we can calculate an area under the curve, and that is then used for quantification in MS2. And this quantification in MS2 can, of course, then be used for quantifying data sets that were acquired using PRM or DIA. We also allow for identifications to be missed. So this is delineated here with a red dot. So if you happen to not identify that particular peptide here in one scan, we allow, I believe, 2 scans to be missed in a series of consecutive scans. And when you make use of this deconvolution approach and the contributions that I mentioned earlier, these are the plots that you can generate from a typical PRM run. The peptide that was targeted was the orange peptide here, but at the same time as we were trying to measure the orange peptide, we co-isolated these 3 other peptides that beautifully elute over retention time, and a typical PRM workflow would never actually identify or quantify them. Chimeras does that out of the box. And it does so with an accuracy that is on par with Skyline. So here on the right-hand side, we took data from a PRM data set where 52 peptides were targeted, and we quantified them with Skyline and with the approach that I mentioned earlier with Chimeras. And as you can see, that basically forms a diagonal, and meaning that the quantification we do with Chimeras is on par with the quantification of Skyline. And with that said, all there is left is basically to discuss, well, is there really a difference between a DDA and a DIA spectrum, or a wide window DDA or wide window acquisition and DIA spectrum? And here I brought you 4 different spectra, and I'm sure you can appreciate, if you look at spectra frequently, that some of these DIA spectra rather look like DDA spectra, and some of these DDA spectra really look like DIA spectra. So Chimeras has the big advantage that it uses the very same approach to analyze all of these different spectra and calculates the same scores and the same figures of merit, so that you can really, for the first time, compare DDA and DIA fairly to one another. This is the typical workflow that we would use. We start with the spectrum files node, as I mentioned, spectrum selector, and then chimeras, and it's one unified workflow for all the different data types. Don't pay attention to the dynamic modifications. Please don't do that. That will take forever. All right. So how does it then perform on DIA data? So on DIA data, when we compare ourselves to other search engines, what we typically do first is we verify that our error rate is under control using entrapment experiments or double decoy experiments. So here in this particular case, we again took a data set from the public domain. It's a two-organism mixture, and we calculate whether the FDR that we calculate is actually the true FDR by spiking in peptides that we know shouldn't be in the sample. But we don't take these peptides from a different organism. We permute the peptides that we expect to be in the sample in such a way that we shuffle their sequence and keep the termini constant. So we generate very, very similar peptides that that shouldn't be in the sample, and we measure whether we are able to accurately distinguish them from true peptides. And in an entrapment plot like this, where you want to be, so you want to be as close to the diagonal as possible. And in fact, Chimeras FDR and entrapment FDR really are on the diagonal, visualizing that we have our entrapment FDR under control. So if we now go ahead and analyze the same data set with Chimeras, Diane, and Spectronaut and ask the question, well, what's the number of precursors that we can identify at 1% FDR or 1% empirical or entrapment FDR, we see that for Chimeras that doesn't change anything, for Spectronaut that doesn't change anything, but for Diane the number of identifications is substantially reduced in comparison to when you cut at FDR. So the takeaway here is that Diane in this particular entrapment experiment setting underestimates its FDR by a factor of about 3.4, I believe. So they claim to have 1%, while they actually have something along the lines of 3.4% FDR. If we look now at quantitative performance, what we see is that the known ratios that were present in this particular sample are well reflected. We here compare again Chimeras, Diane, and Spectronaut to one another and plot typical MA plots, so log 10 mean intensity of a precursor on the x-axis and the fold change on the y. And you see that the quantification of Chimeras in comparison to, for example, Spectronaut is very similar. For Diane, I believe here we didn't use the entrapment data, but the original data. And if you look at this in a different way, what you see is that the ratio density plots, so just quantifying whether you really are capturing the true ratio of Chimeras, are very comparable to Spectronaut, meaning that with the totally different approach of calculating these contributions, we can arrive at very similar quantification as peptide-centric search engines such as Diane and Spectronaut. Chimeras itself is a spectrum-centric search engine. And as such, you can use this property to actually now compare DDA and DIA data from the same sample. So here we compared again from on a data set from the public domain, where the same sample was measured in DDA and in DIA, and we just quantify the number of identifications that Chimeras performs at the PSM and peptide level based on these different raw files. And as you can see, the number of PSMs is far greater in DIA in this particular case, but the number of peptides is greater actually in DDA than in DIA. That might be a function of the fact that this particular study was done on, I believe it was an HFX, so they had to use quite wide isolation windows in DIA, which likely reduces the number of peptides. So we would be very curious how a plot like this looks on a newer instrument, such as, for example, the Astral mass spectrometer. But if you look at the quantification performance between MS2-based quantification and MS1-based quantification, what we see is that our MS2-based quantification and our MS1-based quantification is very similar, meaning that we basically can recapitulate MS1-based quantification using spectrum-centric MS2 readouts. And with that, I probably overwhelmed you with information, but I would like to end in a couple of key takeaways. So CHIMERYS 2.0 will be available in Proteome Discoverer 3.1. It will be able to analyze DDA, DIA, and PRM data. We perform deep learning-based predictions, and hence can analyze these types of data sets irrespective of acquisition method. We perform quantification based on these contributions that I mentioned earlier, and that is basically as accurate as quantification using XICs in Skyline. We perform automatic peak boundary detection as well in PRM. So that is basically allowing you to automatically process PRM data. And for the first time, you now have a tool with Chimeras at hand that allows you to fairly compare DDA and DIA measurements of the same sample to each other. So all in all, I think it's safe to say that we can predict a bright future for machine learning and proteomics. And with that, I'm at the end. I would like to thank the MSAID team. Of course, I'm just the one that presents all of this. There's a whole team of people that make all this happen. I thank the team, the Proteome Discoverer team at Thermo. And of course, if you're interested in Chimeras, you can request a free demo. And if you're a bioinformatician that is interested in working with us, feel free to reach out. We are always looking for talented people. Thank you very much.